So welcome to this seminar, which is a part of a series organised by the Human Developmental Biology Initiative. And today is held prompted and with collaboration from the Gurdon Institute at the University of Cambridge. These seminars are intended as a respectful discussion about sometimes contentious subjects related to developmental and stem cell biology research. In organising this session, we aim to create an open and inclusive and safe environment where people feel welcome to contribute and to participate in discussion. Indeed, we encourage active participation and have allowed plenty of time for questions after the speaker's presentation. So feel free to write questions and comments in the chat, and we'll pick them up as the discussion develops at the end of the talk. If you want to do that anonymously, you can submit them through Slido, and Pilar will provide a link. Please be aware then that only the talk part of this session is going to be recorded and not the discussion. So feel free in the discussion to develop ideas together. So our speaker today, great pleasure to have him with us, is Professor John Evans. He's the Tata Chancellor's Chair in Social Sciences. He's Professor of Sociology and co-director of the Institute of Practical Ethics at the University of California in San Diego. John specializes in examining debates that involve religion and science in the public sphere, as well as using social science approaches to contribute to humanistic and ethical debates. His most recent books are The Human Gene Editing Debate, which was published in 2020 by Oxford University Press, and his current one, which you can see on the screen, Disembodied Brains, Understanding Our Intuitions About Neurochimeras and Human Brain Organoids, which was published last year, 2024. And it's on this topic, obviously, today that John is going to focus, and his title is The US Publics and Professionals' Ethics of Human Brain Organoids and Human-Animal Neural Chimeras. So it's a great pleasure to have you with us today, John. We look forward to your talk.
Thank you very much. It's great to be here, wherever here actually is in a Zoom talk. I'm actually in Edinburgh right now, but it's great to be here. That's the book on the screen. For every book I sell, I make 90 cents towards my retirement fund. If I make $400 over the life of the book, that's a major success nowadays. But I will be talking about this book, and I gave a different title to make it clear that I am talking about the American context, but I do believe that what I'm saying does extend it through the Anglophone context more generally, but also to the UK. Here's what I'm going to cover, believe it or not, in the short amount of time I have today. And wherever you see the color orange is where I am, in case you check out and need to check back in mentally here. So I'm going to skip over some aspects that everyone in this room will know about, such as that's a human brain organoids in the dish. And I think everyone knows how those are created and what people have been doing with them. You know, most on my campus, for example, there's people who are using them to study neurological degeneration disorders. And, you know, at present, they really have not solved the problem that would require solutions to make them bigger than 4 or 5 millimeters, but people are working on it. You know, there's no vascularization. They're one component of the brain. They don't really interact with the environment yet, which is a critical importance in the debates in the philosophy of mind. If you don't interact with anything, you know, that's a different type of entity. However, as I think everyone on this call will be aware, people are working on vascularization, assembloids connecting different organoids to different parts of the brain have been created in various ways. And again, more important for the philosophy of mind is that there are the slightest hints at interaction or experiences. So, you know, you can shine light on cells, retinal cells that are connected to an organoid, and you get a certain response. There is a burgeoning field, at least in the United States, on organoid computing, where you would connect electrical leads to an organoid to create a organic mechanical computer. So this is not really there yet, but underway. So that's an organoid. That is a chimera. Obviously, we don't have any of those, but a chimera is a non-human animal with human cells in it. This is the kind of chimera we're more used to, which is the long history of the humanized mouse, which this one is one of the mice with a human-like immune system used extensively in medical experimentation. So to start to get into the topic of today, I'm not going to talk about friends like our little mouse here. I'm not going to talk about non-neurological chimeras. What I'm interested in is a neurological chimera, a non-human animal with human neural cells in it. And of the ways you could possibly do this, you could get an organoid and surgically implant it into an animal, very commonly done. The Salk Institute, which is about 100 meters from my office, has a lab with lots of mice like this. Blastocyst complementation, where you take a mouse blastocyst, for example, you let it grow to 200 cells or so, and you ablate the part that's going to become the mouse brain. You put human stem cells into that part, and you have a partly humanized mouse. And then obviously there's the idea of genetically modifying animal embryos in various ways. I'm not going to talk about the differences between these technologies because the differences between the methods here lack moral weight from an elite perspective, and also the public's perspective. People don't make a distinction between how you would do this. It's more like that you would do this, okay? And I think everyone knows, and I always try to remind the public when I talk to them, that if you created a mouse with a partly human brain, it's not like the mouse is going to stand up in its cage and say, "Hi, I'm Mickey." It's at best, some experiments have shown that these mice are slightly smarter, quote unquote, by mouse standards, but that's still under a great debate. Mostly this is being done with mice and rats. There is concern that if you can do it with the mouse and rat, you can do it with the non-human primates, and such research is currently underway. So again, why do we do this sort of thing? This is important, especially when I talk to public audiences, which is there's a good reason for doing this, which is to reduce the suffering from neurological disease. These are what are called human brain substitutes. And why do we need a substitute? In one of my favorite lines of all time, it's a great time to be a mouse with Alzheimer's disease, which is all these spectacular treatments that work really well in mice, as I think people on the Zoom call know, don't necessarily work very well when you try to apply them to humans. So we need research on human brains, yet you can't really experiment on live human brains. So what do? You create these substitutes. And that's what this is motivation for here. So let's get to the ethics. There are a number of more, what I will call narrow ethical questions that people are working on that I do think are important. In the public in particular, there is going to be a subset of the population that does not approve of experiments on animals in the first place. I'll simply say, if that's your position, you would have gotten off this train long ago. Okay, so that's just an issue that certain people are concerned with. There's also a lot of concern with the level of consent that would be required to grow an organoid from someone's tissue. A lot of the cell banks that exist, at minimum, I would say that the people have not given specific permission to grow a human brain organoid, to say nothing of having such a tissue installed in a non-human animal. And so what sort of consent do you need? Do you actually have to go reconsent all those people? There's a lot of debate about that. More importantly, if you create something like a mouse that is as smart as a pig, do you have to give it a more pig-like environment? The different standards for laboratory animals of various sorts would require changing the environment, and then ultimately, at a certain point, you couldn't experiment on them anymore if they became too human-like, quote unquote. And then what are the evidentiary standards for societal safety? I think that's another big question, but I want to focus elsewhere today. We should start by asking whose ethics should determine what research occurs. I'm comparing in this talk and in that book the ethical views of professional ethicists with the public. And the reason I compare with the professional ethicists is they have the most influence over what actually happens with policy in the United States. In the UK, if I were doing this, I would have, examined the people who've interacted with the Nuffield Council or something like that. But there's sort of US analogs to this. So in the rest of my talk, I'm going to be comparing the ethical concerns of this academic bioethical debate with the views of the public. So how do I empirically determine what academics believe? That's incredibly easy, given that they write it all down in exquisite detail and publish it in journal articles and appears on the internet. That's fairly easy. The public is a bit more of a challenge. The larger project here, this is all based in sociology. There were 35 in-depth interviews with a stratified sample of the public, stratified by critical demographic characteristics that I would expect would influence people's views of this view. Most importantly, in the US context, and I think in the British context as well, the most critical demographic variable of views of issues like this is education level. So if your sample is everyone has an undergraduate degree, you're going to produce a wildly biased conclusion. So, and then the US, a bit less than Britain, is you need to have the right religious representation. And so I have a third conservative Protestants, 20% liberal Protestants, 33% Catholics, and the rest, non-religious. So the one thing to say about research like this, just so you're aware, is I tend to specialize in research in these deep abstract questions. There's typically not one test that you can do to come up with a definitive answer. Instead, I think of it as like painting a mosaic for you, where in the end, if I'm painting this picture, you'll be able to tell that it's a dog and not a cat. It will not show you the details of the whiskers of the animals. And that's just the way this sort of research goes. Okay. Now, so I also want to say at this point that while this research was done in the US, I based a lot of the theoretical expectations of what I would find on British research. And so the British social science community has long been concerned with issues like this and I expect I would find something similar in the British public. So with that all said, what did I actually do? So for this, I use what's called an experimental design vignette study. This has a number of advantages when it comes to investigating these abstract ethical issues. The idea is to use randomization to avoid confounding factors. It's very much like a clinical trial. So for that COVID vaccine trial, we would worry that the vaccine worked because of the racial background of the trial participant or the education level or something. But we don't worry about that because we randomly assign people to the treatment or control arms. So if fewer people from the treatment arm get sick, we can conclude that it's not due to any characteristics of the respondent, but due to the treatment itself. Well, the vignette survey does the exact same thing. A vignette is a paragraph-long description in a survey where survey respondents are randomly assigned to see particular chunks of text. At the end of the vignette, the respondent is typically asked, do you approve of this technology? The key is here, just to kind of go here, that the respondent doesn't know what the other people are seeing, so they can't game their responses. This is a major problem in social science research, is people trying to anticipate what the researcher wants and then telling that researcher that same thing. And so you used to use methods to avoid people doing that sort of thing. So my vignette with organoids started with a human brain organoid is a 1/2 inch or 5 inch. So half the respondents saw 1/2 inch, half saw 5-inch. And yes, I recognize there are no 5-inch organoids. This is a sort of strategy of taking extreme cases to generate distinctions, moral distinctions. And we could talk later on about, the tension between creating questions about technologies that do not and possibly cannot exist versus what I've done here. And then the question is, do the respondents who saw the half-inch version of the vignette respond differently to those who saw the 5-inch version? If so, we can assume that there's something about the size of the organoid that generates people's response, okay? Because they don't know that the other people were seeing something else. So there's a number of other features. There were 64 possible descriptions the respondent could have been randomly assigned to see. So it looks like this. This is how you're presented in an academic journal article. This was the example. What you see is underlined. They didn't see underline. That's just for you, okay? And those underlined parts are the ones that varied. So I'll let you read that really quickly. So half the respondents saw that one had been created, half saw that 10,000 were created. Half the respondents saw that they were grown, half saw manufactured. Half saw the debate was about whether they would obtain self-awareness, half saw awareness of its surroundings, which is a distinction in the philosophy of mind I won't get into here. And I don't have time to talk about all these, but I'm going to talk about the last one, which is what its level of consciousness may be. And in this example, it could obtain the level of consciousness of an insect. So each respondent only sees one of these, and they're not aware of the other possible pieces of text. So after this vignette, the respondent is asked their agreement with the statement, research into human brain organoids should continue, sort of an agree-disagree scale. So to start with the end of the story, averaging, think of it as averaging across all of the possible 64 vignettes, there is very strong support for human brain organoid research in the United States. And, in case it's not obvious, I would have gotten very much different results if I hadn't included in the end that the purpose of this is to generate treatments for things like Parkinson's and Alzheimer's disease. But that is accurately what the research is about. And that's how it'll eventually be presented to the public. And so this is the best we can do. And with that in mind, there's strongly agree. Number 4 there, which is a midway point between the neutral position and strongly agree, is the modal response. Okay, I also created a vignette for the neurochimeras, and that is that one. So half the respondents saw subspecies, half saw a dozen. Half saw monkey, half saw mouse. There are 4 different versions of how the animal is changed, including one showed here about tissue implant. And a third saw what are described as a humanized mouse or monkey, whichever, to make it correspond with the other part of the vignette. A third saw chimera, and third saw sea beer. And before you wonder about my advanced biological training, where I'm coming up with a life form you're not aware of, I actually made up the word sea beer, okay, because it is a term that has no moral baggage attached to it, just to see what the sort of neutral response would be. All these things are representing debates in philosophy, bioethics, academic theology, and that sort of thing, which I won't get into, but that's all in the book. Right after this, I ask if they strongly disagree, etc., with the statement, I support the creation of these chimeras. Again, the empirical question is whether you get more agreement from respondents who are randomly assigned to see the different versions. So just to get our minds going on this, to tell you the obvious, the people who saw monkey instead of mouse were much more opposed to this research. So, I think that's pretty clear from what we know about public opinion. Now, here you get a lot more mixed responses with a tendency towards, agreement, but with a lot of disagreement. So the question I'm going to be answering, and the typical sociological question, is who is more on the disagreeing end of this, and who is more on the agreeing end of this? Okay, so that's just the introduction to the survey debate. I don't have much of the interview data to show today due to lack of time. Let me turn to the expert ethical debate. So let's start with the concern of people in the United States who are called bioethicists. In Britain, they're often called bioethicists, but often will go by other terms as well. The first media stories of these technologies quote bioethicists who state their concerns that organoids and chimeras will become human, quote unquote. Now, no one obviously believes that this would become a biological human. What they mean there is sort of a philosophical human, which is that they would obtain the same moral status as humans. They would not be physical humans, obviously, but moral humans. This reflects one of the assumptions in bioethics, which is their theory of what a human is, which is that human is that which has certain capacities. Now, I know many people can't imagine what another version is, but in previous books, I've shown that the public's version of what a human is quite distinct from that. So, with the idea that humans are those who have certain capacities, if you lack these capacities, you have different moral status. So human fetuses have a different moral status, and therefore abortion would be acceptable. People who are permanently comatose lack certain capacities, and therefore ending the life of comatose persons is also acceptable. Anyone who's had an advanced medical directive is familiar with this logic. Without certain capacities, I do not want to continue living. So this is the sort of reasoning that's very common. It's the sort of domain assumption of bioethicists, at least in the United States. So some animals like chimpanzees have a lot of these valued capacities and thus should be treated at least more like we treat humans, or perhaps equal to humans, and better than the biological humans who lack these critical capacities. So therefore, an organoid or a neurochimera would obtain the moral status of a human if it obtained enough capacities that a human has, most notably in this debate, consciousness, which is like a threshold condition for moral capacities. So for example, in the news article describing this mouse that a human brain organoid implanted in it, Hank Greeley stated that the question was whether you're creating something human-ish that you have to take seriously in terms of according dignity and respect, and figuring out what that even means. Another bioethicist states that if we give them human cerebral organoids, what does that do to their intelligence, their level of consciousness, even their species identity? The animals might become too human. Greeley sets the standard, says whether you could transform a mouse into a man, but whether it was a creature with some aspects of human consciousness or some distinctly human cognitive abilities. I'll let you read the quote by Chen. So this notion of consciousness is essentially the only question that's considered morally relevant. This is the charge given to the National Academy's commission on this thing, and for Truth in Advertising, I was a member of that commission. You'll note that the red is a statement about consciousness. These are what we were supposed to figure out, and it was just all about human awareness, et cetera. Will these animals and organoids reach that level? That's what the debate, at least in the US, is about. Okay. So what is consciousness? I'm not going to tell you. I mean, this is, to my mind, the largest morass in the academic world. You could spend your entire career, get your PhD in philosophy of mind. You will not definitively answer this question. And again, everyone in this debate recognizes the incredible difficulty you would ever have in determining whether or not any animal has levels of consciousness, et cetera. And I know that there's the mirror test and that kind of thing, but it is at best rudimentary. So whatever it is, we can all agree that insects have very little of it. Mammals have more, and humans have the highest level of it. Obviously because it's not mice that set these standards, we did. And so therefore, we're on the top of this continuum. Okay. So this is what bioethicists believe is the moral problem. The moral problem is, would a neurochimeric animal like a rat, or most notably a non-human primate, obtain something more like consciousness? So after the organoid vignette, I asked whether they approve of the research. And I also asked whether the research is done, the organoids could be destroyed, which is a good indicator of moral status. In the bioethics arguments, more consciousness means more moral status. So the question is, did the people who saw slightly different vignettes have different responses to these questions? So a quarter saw an organoid could obtain the consciousness of an insect, a quarter saw a pig, a quarter saw a human fetus. And this is what I've called predicted values. I'll skip that in a second. But at first here, the percent who approve the research and the percent who agree it's okay to destroy the organoid for no consciousness, which is the baseline condition. Okay. But when you look at the other levels of consciousness, there are really no differences. So this is a not statistically significant difference. No matter how you describe the level of consciousness, there's equal willingness to destroy the organoid, for example. So if people were really concerned about consciousness, you would see an ascending pattern here as you go down the chart. Let me turn to neurochimeras. I asked, should we treat these things better than we treat other, mice or monkeys, whichever one they saw? This gets at the moral status of the entity. There are three that they could have been randomly assigned to see. The animal having a human immune system, that's the baseline comparison. Brain tissue from a human, brain tissue like a human. And if consciousness matters, you would see, again, differences. And these differences here are also not statistically significant. So let me turn to chimeras for a while. The public concern is different for organoids and chimeras. So when social scientists hear of a neurochimera, their thoughts don't turn to consciousness. Knowledge of Western cultures would suggest that the main ethical concern is about a foundational divide between humans and animals. Let me explain. Societies have these foundational cultural distinctions that are so deeply assumed that they're difficult for people to see. So in the West, distinctions are man, woman, mind, body, nature, culture, human object, and of course, human animal. And this quote from Smits summarizes, you can read faster than I can talk. So how do you know when something is violating one of these distinctions? It's because it becomes the subject of fear, disgust, and fascination. Okay? So people often think that only, primitive societies hold these sorts of binary beliefs, but in actuality, all societies do. They're just different. And I should say that people can train themselves to not be bothered by these distinctions. Like classic studies in medical sociology show that the inside-outside the body distinction, which all cultures seem to have, the point of the anatomy lab in medical school isn't to teach you about the inside parts of the human body. It's to have you get over this inside-outside distinction so you can be an effective doctor. So how do we know if people have these distinctions? So here's a common reaction. That's the plastinate on the left, and that's a zombie on the right. Both of them that are violating a distinction. The left one is violating the inside-outside, and that usually gives people a sense of disgust. And on the right side, the reason that people have been fascinated with zombies all this time is it violates the sort of living dead distinction. This, of course, reinforced by culture. Who's that? Our most famous medical doctor of all time created Mr. Frankenstein here, Dr. Frankenstein's monster, which also violates the living dead distinction. This becomes part of popular culture. Many of you have seen Planet of the Apes, and I wonder whether or not British people have avoided being subjected to the one on the right, which is Mr. Ed the Talking Horse, one of the worst shows of my youth. It's amazing that we watch such things. But anyway, Mr. Ed had human-like capacities. And he could talk, but he could only talk to his owner, Wilbur. Many people have a reaction to this sort of a sense of disgust because this is an artist's rendition of a humanized pig, but the idea is to sort of mix the two, which shows you this response. Okay, so I'm not the first who've noticed this. Many people have talked about that this connection would cause concern for the public. So a critical moral problem then becomes, does a neurochimeric animal violate this distinction between humans and animals? Does it actually exist in the public? So I did some non-vignette survey questions here. And so one of the questions was, in general, humans are more similar to animals than they are different. This is a measure of the extent to which people see a divide between humans and other animals. This chart shows the percent of those who see a human-animal divide who approve of neurochimeric research. In the next row, those who at least agree with the divide, So what we see here is those who most agree with this divide are the least approving of this research. And those who are least agreeing with the divide are most approving. There's a relationship here between seeing this divide and approval of this neurochimeric research. This is not as big a difference, but it is statistically significant. Similarly, it is those who do not see a foundational divide. What we see here is, I'll give you this. I remember I told you I named these things differently, what called a humanized, a chimera, or a sea-beer, okay? And what is the different levels of approval for me labeling this entity in certain ways? Well, when it's labeled as humanized, okay, you get 30%, you get the same for chimera, but when it's a sea-beer, many more people approve of the research. Why? I would argue that humanized reminds people of the human connection to this entity. You're sort of labeling it as partly human, and therefore you're generating this negative response. So to sum this section up, members of the public who see a foundational divide between humans and animals are more likely to oppose neurochimeric research. And since a very large fraction of the public believes in such a divide, this is one of the explanations for a lack of support within the United States, at least, for neurochimeric research. And I think you would find something similar in the UK. So let's turn to organoids. Again, in bioethical debate and the scientific community, disembodied human tissue has no moral status. Okay, if you had a tumor removed from your body, it's not treated as sacred in any way and is often burned as medical waste. But again, the public has a different view of this. There's been studies of people's disembodied parts. This is a British study about blood donation. Half people believed their blood was a separable thing, but some believed they sort of had a continuity of position, like a perpetual loan. Some believed that their blood was linked like a parent would to a child, and many people believed that their blood became the bearer of a personality or moral disposition. There's a stronger version of this, which is almost communicative, this notion of personality. Classic studies of tissue donation show that the physical nature of biospecimens retains an individual's essence. So people will say things like, I feel more uncomfortable with giving a biological specimen because it feels like an attachment or extension of myself. Classic studies of organ donation show that people think that the organ retains the essence of its originator, okay? And many studies, including Haddow here in Edinburgh, find the same thing. And I'll just say that the truth of this matter doesn't matter, okay? The fact that people think this shows you where their mind is on things like this. You could eventually show people that know that liver does not retain the essence of the original human, but people's original first instincts are that it does. So does this concern exist with the public? So a organoid is like disembodied human tissue. It's grown from an existing human's cells. So from a scientific and ethical, bioethical perspective, it's just 4 millimeter lump of tissue in a dish. But I think it's something more for the public. I asked, human brain organoids should be thought of as an extension of the person who donated the skin cells used to make it. You have more people agreeing with that than disagreeing with that. More extreme, if a human brain organoid could think, it might have the thoughts of the person who donated the skin cells. Even stronger view of that. Now, again, this is obviously scientifically ridiculous, but we have to go with what the public's original impulses are. And the idea is that disembodied human tissue has this ephemeral connection to the person who gave the tissue. So I look at the extent of agreement with destroying organoids or whether they should be treated better. The comparison on the left is with agreeing or disagreeing that an organoid is an extension and agreeing or disagreeing that it has the thoughts of the donor. So look at the left column at the top. Again, people who think that the organoid is an extension of the donor are much less likely. They agree with destroying the organoid, give it less moral status. And the people who disagree that an organoid is an extension of the donor, which is the more scientific view, agree with destroying organoids. And you have a similar, albeit not as large, situation below. It's even more extreme there, and yes, that 4 there is not an artifact. I probably checked that 50 million times before publishing the book. I can tell you the statistical story about that. But you have the same story here. It's the people who think that organoids are an extension somehow of an existing human are the ones who are less supportive of the research. So in the interest of time, I'll just simply say that recently I was on a National Science Foundation, which yes, it does still exist apparently, National Science Foundation panel deciding on research projects for this organoid computing thing. And I'll simply say that the idea of getting a bunch of organoids and using them to develop research to heal neurological disease, I think most Americans will say, that makes me uncomfortable, but I'm willing to do it because neurological disease is this terrible thing I'd like to have eradicated. If you say, I'm going to create human brain organoids and wire them up into a bank of computers in order to create faster computing power for artificial intelligence applications, I think you're going to have very, very low support and opposition growing from that sort of thing because there isn't a quote unquote good reason for doing so. So we'll skip over that. Let's go. So I have time for questions. Let's go to the conclusion. I've summarized this ethical debate about organoids and neurochimeric animals. For the academic bioethicist, the most important consideration is whether either of these could obtain enough consciousness to warrant different treatment, or as extreme, treatment like a human. My survey suggests that possible consciousness is not very critical for the public's view of either organoids or neurochimeric animals. Secondly, the public tends to adhere to a foundational distinction between humans and animals that academics have largely learned not to be concerned with. The survey suggests that this divide is important and a reason for opposition to neurochimeric animals. I'll simply add a little autoethnographic statement here, which is when I finished my PhD, I was doing other sorts of research for a while. When I started entering this field, I initially had the same sort of reaction that the public did. But by sitting through many, many scientific meetings where people talking about modifying animals in various ways. I too sort of became used to this, and I think sort of taking on the view of scientists and ethicists by exposure. So I think this is one of the reasons why basically people with PhDs have a different view than the average member of the public. Third, the public sees possible connections between disembodied human parts and actual humans. And this connection gives a degree of reluctance to human brain organoid research. Again, when you do the in-depth interviews on this, the public is also very concerned about people suffering from neurological disease and support research that reduce the suffering. Thus, it's an actual ethical dilemma. But as this research goes forward, I think we need to be aware of the reasons for the public's ethical concern so that research can advance in a way that's most consistent with the public's ethics, that is both more democratic and will ultimately result, in my opinion, in more trust in science in this area. So with that, I will stop and answer any questions you have.
Thank you very much, John. That's very stimulating discussion. Well, very stimulating ideas there.